Okay, looks like it's 4 o'clock, folks. Okay, we're going to go ahead and get started. I'm going to introduce our speaker. It's my sincere pleasure to be able to welcome our speaker today, Dr. Travis Heggie. I've been wanting to welcome him here since the spring when he was going to be visiting in person, but then, as we all know, we went to remote and the seminar series was canceled for the rest of the semester. And then he was also scheduled earlier this semester, but we've experienced these storms coming in on a fairly regular basis, it seems like, this semester, so we postponed to now. So finally, we have Dr. Heggie here to present his research. So just a little bit about him. He's at the Mississippi University for Women as an assistant professor. He's been there for just a couple years. Before that, PhD at University of Idaho, and then postdoctoral work at both Idaho and Michigan State University, which is one of my old stomping grounds. So he has 3 main areas of research. So one of which is looking at how morphologies determine what organisms can or can't do, basically. Then he's also looking at macroevolution of traits, specifically when they arose, and then over macroevolutionary time, how they have changed. And then finally, matching species traits to their environments to study aspects of adaptation, basically. And he's working on some cool organisms, geckos. So I'm really interested to hear what he has to say. And he's got some cool videos on his website if you're interested in such things. So the other thing I wanted to add is that Dr. Heggie is a really big advocate of STEM education and outreach. And so he has a really active accumulation, I should say, of times when he's gone out to get his science out to the public. So doing research activities. And so that's fairly unusual and actually really cool. More researchers should be doing more of that. So with that, I'm going to turn it over to Dr. Heggie, so you can share your screen and take it away.
Cool. Thank you very much for the introduction. Let's see, everybody can see everything, right? Hopefully, yep. Everybody can hear me. Cool. Yeah, so as I was putting this talk together, I realized besides the couple times that we had to reschedule everything, one of the fun parts of coming and doing kind of these in-person departmental seminars is you get to spend kind of the round robin 30 minutes with everybody. And so that's the part I was kind of bummed I was going to miss, was getting to hear 30-minute pieces of what everybody else in the department's doing. So that's kind of the biggest thing that I was disappointed with, is that I didn't get to learn what everybody else is doing. But yep, I thought that was a great introduction. I'm going to be talking about geckos. I'm over at the Mississippi University for Women. And so we'll go ahead and get started with that. And so my research in a really general sense pretty much asks, why are animals shaped the way they're shaped? And so we can see there's a bunch of cool different kinds of lizards here. And so some lizards have long legs and they're sprinters. Some lizards are little thick tanks. Some lizards are cryptic. Some lizards have googly eyes that let them see in multiple directions. And so like the introduction said, I study geckos. And so geckos are a really cool group because they're super diverse. They're one of the oldest groups of lizards. They're 200 million years old, depending on who you ask. And they live on almost every continent except Antarctica. And you can find them in tons of different environments. And so they live in tropics, they live in deserts, they live in temperate environments. We tend to think of geckos as, thinking like the Gecko gecko, right? As climbers with sticky toes. And lots of geckos do climb trees and climb rocks, but there are lots of terrestrial groups out there too. And there's actually whole groups of geckos that don't have toe pads. And so the morphological diversity is really big. Variation in body size. So some geckos are really large. Some of the smallest vertebrates are geckos. And so this is an adult spherodactylid gecko that's only the size of a dime. And so there's lots of really cool adaptation and diversity. And so there's lots of really cool questions we can ask. And so I specifically think about their toe pads a lot. And I kind of can frame my questions into just generally asking, what's the diversity out there? How are their toe pads shaped? Where did they come from? Thinking about macroevolutionary patterns, origins of these different traits, and then trying to connect that to biomechanics. How do they actually work? What's the engineering and the physics behind them? Because gecko toe pads are a really cool example of natural nanotech. And there's actually lots of engineers out there working to try to mimic it, which is kind of fun. And then we can bring in the evolution and the adaptation, trying to think about, given the morphological diversity we see, and given what we think the mechanics are underneath them, how is that helpful? How is that useful? What are the kind of the adaptive patterns that we can see? And so that's kind of the different arms of my research, and some projects combine some of these ideas. And so I'm going to talk about a handful of different projects I've been working on the last couple of years. And so to kind of start off, some of you might not be super familiar with Gecko Toe Pads, and I know that there's a handful of undergrads in here too. And so I wanted to start by thinking about, can we list all the different type of human-made adhesives? And so either in chat or yell it out, can we come up with what are all the different kinds of human-made ways that we've invented to stick stuff together?
Glue.
Glue.
Tape.
What's that?
Tape.
Tape. So tape is in essence glue with a backing, right? Rubber cement, which is another just form of glue, right?
Does Velcro count?
Velcro. Think about permanently attaching things and temporarily attaching things. So we've got different forms of glue. We've got Velcro. Nails, screws, mechanical connections, right? Buttons, zippers, snaps.
Staples.
What's that?
Staples.
Staples is another kind of mechanical attachment, right?
You must be missing a good one.
For an answer.
How do you stick stuff to smooth glass?
Suction cups.
Suction cups, welding. So welding is another one. So welding is actually melting things and permanently combining them, right? And then there's kind of one more big one that only works with metal. Magnets, right? So we've come up with lots of different tools that work in lots of different scenarios, right? Generally, different types of glues and tape are kind of more permanent than not, but they work on a variety of surfaces. They work on if the surface is rough or smooth. Suction cups kind of only work if the surface is really smooth. And for suction cups, you need air pressure behind them, right? Suction cups wouldn't work in a vacuum. Velcro, you need two different types of surfaces that work together. Snaps, you've got to start modifying things. Nails, staples, actually you have to change the materials, right? Magnets only work on metal. And so out of all of this, what do we think Gecko Toe Pads are most similar to? Does anybody have any hypotheses?
Suction cup.
Yeah. Velcro.
So a lot of people go Velcro or suction cups, right? Those are the two most common intuitions. And actually Gecko Toe Pads at a molecular level actually work most similar as the magnets, which is really weird, right? Magnets, exactly. They're most similar to magnets, which is super interesting. And so what actually happens, so if we zoom in on the bottom of a Gecko Toe Pad, their bottom of the toe pads are divided into specialized scales, and each individual scale is covered in tiny hair-like structures. And so these are reptiles, they're not mammals. So they don't have a hair that's embedded in their skin like we do. These are just extensions of the outer layer of their skin. And so their skin is mostly beta keratin, which is a protein. Our hair and our fingernails are mostly alpha keratin, a really similar protein. Bird feathers, snake skin is all beta keratin. So it's a fairly hard material. If you think how stiff your hair is or how rigid your fingernails are, it's a tough material. It's a tough biological material. And so these hairs, if we keep zooming in, so here's a human sperm cell and a red blood cell and E. coli that's kind of just pasted in this picture to scale. So these hairs are, depending on what type of gecko you look at, maybe 100 microns tall. So they're really small. They're less tall than our hair is wide, depending on what kind of hair you have. And these tiny hair-like structures are branched. So if we keep zooming in, they're branched and they're curved backwards. They're curved towards the gecko's palm. And then if we can keep zooming in even farther, each individual tip of each individual hair is flattened out like a spatula. And they're actually called spatulae. And so these tiny little spatula structures are only 200 nanometers wide. And so does anybody remember the wavelengths of light that our eyes can see? Nobody?
What do you mean?
470 to 680 or so.
There you go, 400, 600 nanometers, right? So if these tiny tips of these hairs are only 200 nanometers wide, they're actually smaller than a wavelength of light that our eye can see. So they're incredibly small. And so what actually happens when a gecko plants its foot, and pulls, these tiny spatula-like structures can get close enough to whatever the gecko's walking on that the electrons that are spinning around the proteins start to push and pull on the electrons that are spinning around the surface, spinning around the molecules that make up the surface. And so this electron cloud push and pull is van der Waals forces. And so that actually creates a weak attractive force. So it's actually electron clouds that start to interact loosely similar to magnets that actually let geckos stick to stuff, which is super cool. And so there's a couple different ways we can measure how sticky a gecko is. We can take a chunk of these hairs off the bottom of their toes, strap them to some force sensors and some servo motors, and actually pull them across the surface. And so what happens when we do that, so here this is kind of mimicking a gecko planting its feet and pulling. And so those hairs, which are curved backwards, here's an individual hair. You guys can all see my mouse, right?
Yep.
Cool. So here's an individual hair. It's curved backwards. Here's the branching. And then all the tips are attracted to the surface. So those hairs are put under tension and they straighten out. And so they actually act like a spring. So as long as they're kept in tension, they pull up and away at a particular angle from the surface. And so that's what generates friction and negative normal force or adhesion. So we can directly measure how much friction they produce and how much they pull upwards on the surface. And then what's cool is when a gecko wants to detach or lift its foot off, all it has to do is eliminate that tension on the hairs, and the hairs curve back to their natural shape and the gecko can lift its foot off. So it's easily reversible, but it's also really strong. And it works on rough surfaces because all these hairs can operate independently, doesn't damage the surface. We don't have any adhesives that work that way, that can do all this. And what's really cool is the hairs can self-clean. So if you get debris, you know how you have, if you have like sand in carpeting and you run your hand over the sand, it kind of if you run your hand over the carpeting, it actively ejects the sand. These hairs can kind of operate the same way, where if they get contaminated, they actively shed contaminants. And geckos regrow this whole system every time they shed their skin. And so we don't have any adhesives that work this way. So it's a really cool system to try to mimic. The challenge is that we don't have any manufacturing tools that work at this scale. We don't typically design things at a nano scale or even a micro scale. So there's a lot of engineers trying to figure out how to recreate these intricate structures at this really small scale. On top of that, another way we can try to study this stuff is to just plop a gecko on a force sensor, build them a little harness, and just pull them across the surface. And we can measure how much friction a whole animal produces, how much friction they just produce with their front feet, or one foot or one toe. We can do more whole animal studies that way. Another way we can do it is this is one of the cool videos I have on my website. This is some research I did when I was in grad school. So this is a live, not sedated, slightly angry gecko, and he's holding up all of his weight with one single back toe on a very clean piece of glass, as that glass is now turning upside down. And so that'll replay there. And so this whole genus of geckos doesn't have any claws. So that's pure toe pad doing this on smooth glass. And so the really cool part of this test is that the longer they ride that glass, the less friction they're producing and the more adhesion they have to produce, right? The angle that they're pulling at changes. And so the maximum angle they hit tells us about the ratio of friction to adhesion they're able to produce, because we can pretty much just take the tangent of that angle. And so the other thing that's neat about this is that we've put little backpacks on them to artificially make them weigh more. Because you might think this might be weight dependent, right? Heavier geckos might peel earlier because that's kind of the way tape works. but this is a weight independent process. So it doesn't matter how much a gecko weighs, they still always fall off at the same angle. And different species fall off at different angles. So it's really a cool test, and it's probably related to exactly how those hairs are shaped. I'll let the video play one more time.
Poor guy.
Yes. So I've done that for a whole bunch of different species in the lab. I've actually built field versions of this to take in the field. And so, yeah, so that's a kind of a fun performance test to do with them. And so now we've got kind of an introduction to a little bit of how toe pads work. So then I want to talk about kind of the general morphological diversity. And so just within geckos, the toe pad diversity is crazy. There's tons of different types of toe pads. We might think of this being your generic gecko toe, but there's actually a ton of different crazy shapes and varieties. And we actually see these different shapes pop up at different places in the gecko family tree. So there's 7 families of geckos. There's one whole family that is completely limbless. They're convergent with snakes. They don't have any arms or legs. They're only found in Australia, so they don't have any toe pads. There's 2 more families of geckos that are completely padless and probably never had pads, depending on who you ask, depending on which ancestral state reconstruction you do. They probably never had pads. And then there's lots of examples of gains and losses within the other 4 families that have pads. But then we see these similar shapes popping up in different genera in different families. And yet sister genera might have totally different pads. And so there's a ton of convergence and divergence, and we're not really sure what's driving these patterns. And so one idea is convergence, right? And so this is an Odura gecko from Australia. This is what their pads look like. This is a hemidactylus gecko from the Mediterranean. I bet you probably have these in Louisiana because I know I have them here. And so they're the invasive Mediterranean house gecko, right? And so their pads look roughly like this. If you compare these two, they're pretty similar. And yet these geckos are in totally different families from different continents. Sure, they're both climbers, but that's about that's where kind of the generic similarities, that's all we have, right? Odura geckos live on trees and rocks. Hemidactylus geckos live on trees and rocks. We don't have much more ecological data beyond that. The tough part is that these aren't anoles, right? We don't have really precise habitat use measurements for most species out there because they haven't been as well studied as anoles. And so we have kind of generic definitions of habitat use for them. And so we might be wondering, is this toe pad shape good at doing something? Good at doing something out in the wild? Or another idea is that this might be historical contingency, where if these 2 groups developed pads in a similar way, this might just be the toe pad shape that they got stuck with. And that's why we see similar shapes showing up multiple times in different families. Another cool thing is geckos aren't the only ones with these type of toe pads. Anole lizards, which are a totally different group of lizards in a totally different part of the family tree, also have fairly similar toe pads. They have tiny hair-like structures, but in anoles, the hairs are a lot shorter and they aren't branched. They're just a single stalk, but generally pretty similar. And I know you have anoles there too, you're right? You probably have both brown and green anoles in Louisiana.
Yes.
And so then we can start to ask questions about anoles versus geckos too, which is really cool. And then if we start zooming in again and looking at the hair structures within geckos or geckos versus anoles, the hairs are all shaped different. And so some geckos have skinny hairs that are really densely packed. Some geckos have hairs that are set up into quads, set up in sets of 4. Some geckos have really skinny hairs that are really sparsely packed. Some geckos have hairs that are really thick. So what's the mechanics driving all this? Some geckos have really tiny hairs. We have no idea what the mechanics of the hairs are. We just know that they generally work by bending and stretching. Then there's been 2 different studies out there that have looked at hairs within a toe. And so hair shape changes depending on where you look at in an individual's toe. So as you move towards the tip of their toe, towards their claw, the hairs get thinner, longer, and more densely packed. But also within each subsection of the toe, you see a similar pattern like that. So from an engineering point of view, this looks very functional, right? But what's the reasoning behind it? We totally have no idea. So there's a really ton of cool unanswered questions about just what are the patterns and why are they there, right? And so some hypotheses might be, well, these different toe pad shapes might work better in different microhabitats or something. But again, that might be an example of historical contingency, because we're not even sure. Gecko toe pads probably evolved multiple times within geckos, but again, it depends on how you do in your ancestral state reconstruction, dictates how many origins you get. So until we get a better tool, it's really tough to tell who had pads and who never had pads and who lost pads, who used to have them and then lost them. And on the flip side, we can compare species that are pretty similar. And so this is a Phelsuma gecko. It's a day gecko from Madagascar. All of the species in this genus are diurnal. They're all tropical. They're all arboreal. And they all roughly have toe pads that look generally like this. A closely related genus from Africa, you can see that their toe pads are maybe a different version of a similar pad, but they're all terrestrial rock dwelling. And so we can see maybe divergence from some sort of shared ancestor. And so we see examples of divergence, examples of convergence. It's really cool and really confusing just because we have no idea what's going on. And so I want to talk about, the first project I want to talk about is trying to zoom in on this, just getting a handle on the diversity, period. And so this is a project that I had 3 different undergrads working on last year, and all 3 of them are in grad school now, which is super awesome. And so we use geometric morphometrics. And so we did this for 79 different species. We included geckos, anoles, and a couple species of skinks. So skinks you don't typically think of as having toe pads, but there's actually 4 different species of skinks from Papua New Guinea that have something that if you squint kind of looks like toe pads. So we measured their toe pad, the shape of their toes also. So we use geometric morphometrics, which is a tool that lets you pretty much outline 2-dimensional shapes or 3-dimensional shapes, and then you can start to compare those shapes. So we did this for a bunch of different species, and the analyses for geometric morphometrics is kind of weird because each individual landmark is kind of its own measurement, but at the same time, you want to compare all the measurements working together to represent shape of the whole thing. So it's a highly dimensional data set. And so you kind of analyze your shape information, looking for general patterns across it. And so we do something looking at principal component analyses. And so here's a picture of just some of the example toe pads we were looking at. So here's a gecko, here's an anole, and here's some of the skink toe pads over here. And so we can break everything down into its principal components. And so we can pretty much take our multi-dimensional data set and spin it around to try to find what's the most descriptive dimensions of our shape. So what aspects of shape capture the most info for our data set. And so what we found was this. So we kind of have colored all these different species based on the categories of toe pads we would assign them to a priori. And so the most kind of informative dimension we found first, our PC1, our principal component 1, is in essence a indication of how much of their toe is covered in toe pad and where the toe pad sits on their toe. Is the toe pad closer to the base of their finger, closer to their palm, and kind of narrow? And so there's a couple different species that have this narrow toe pad type shape. And intuition might be that maybe this is kind of an intermediate ancestral toe pad, maybe, right? Without trying to put too much emphasis on what we think this will eventually evolve into. Versus on the other extreme of that axis, we have groups where the toe pad is really pushed towards the end of their toe, really far away from their palm. And so that's kind of one really descriptive axis of variation. The second most descriptive axis of variation is, in essence, again, how wide their toe pad is and where it sits on the toe. So we've got some species where their whole toe is, in essence, their toe pad, and they don't have any toe sticking up, no claw or really highly reduced claw up above their pad, something like these Phelsuma day geckos. Whereas at the other end of that extreme, we have species with really reduced pads and maybe a lot of toe still sticking, toe and claw sticking up above where the pad is. So I've got a picture of an Anoles segria, your brown anole, to kind of represent that, a really reduced pad. And so these are kind of these two maybe most descriptive axes. But what's cool is the black lines on here represent the phylogeny, or the relationships between species, and the nodes represent predicted ancestral states. And so we can kind of look at this and figure out roughly maybe what was happening in the past. The caveat with this is that this assumes the ancestor of geckos and anoles had toe pads, and then it kind of just estimated what the toe pads look like going forward from there, which we know isn't true. We know the ancestor of geckos and anoles is, in essence, the ancestor of all lizards. So we know that it didn't have toe pads, but we can still draw some interesting conclusions from this. So like here, the gray dots are all of our anoles. And so we can see they kind of sit in their own region of morphospace, suggesting that geckos are kind of fundamentally different. Geckos cover all of this other space, but anoles have kind of their own space. So there aren't any, the interesting thing is there aren't any geckos that have toe pads shaped like anoles or vice versa. They're separate, which is interesting. I would have thought they would have overlapped more because that's kind of what they look like. The other thing we can see is that you have a single line going in and then all the anoles are in here. So anoles are their own independent origin of toe pads, so this isn't surprising, but all the anoles are roughly pretty similar in shape to each other. Whereas if we look at like these orange ones, which are different types of geckos that have just a single pair of pads, we see that type of toe pad come up in different families of geckos. And so we can see all these parallel lines coming out to these orange species. That suggests that these species aren't closely related to each other, is that it's parallel or convergent evolution, that we end up with these similar shapes out here. So again, what's driving, is this convergent adaptation? Is this ancestral kind of idiosyncratic historical things they ended up maybe taking the same developmental pathways to grow toe pads, to develop toe pads, and they're stuck in the same spot? It's really interesting. I'm not sure what's driving some of these patterns. And then on the other side, on the flip side, we can see closely related species, but that are fairly divergent in morphospace. And so closely related things are really different. Distantly related things can be really similar. There's a lot of cool questions. Cool. So we've kind of covered generally trying to explore how things are shaped. And then another project I had an undergrad working on was trying to connect shape and adaptation. And so this was a project Bailey Howell worked on when she was a student with me. She's now graduated and she's a graduate student at Virginia Tech working with Joseph Weta, which is awesome. And so when she was here, we were borrowing data from Kristen Witchell, who's a postdoc in Jonathan Lassos' lab, who's done a ton of work with Puerto Rican anoles. And so Kristen was nice enough to share some of her data with us and collaborate with us, looking at Anoles chrysatellis, which is from Puerto Rico. They're a trunk ground anole, if you're familiar with the anole ecomorphs. And so they're pretty much an anole species that lives on the lower half of the trunks of trees, jumps down to the ground every once in a while, but kind of a big, beefy, chunky anole for an anole. The interesting thing about this species is that you can also find them in cities, and the populations that live in town are actually starting to adapt to that different environment. Kristen has done all sorts of work looking at heritability and the different traits that urban anoles exhibit, urban populations have versus presumably their forest ancestors that still have the ancestral state. And so these urban anoles tend to have longer legs, they tend to have different thermal capabilities, and they have bigger toe pads, presumably because the surfaces they're encountering are smoother in urbanized environments, right? Painted concrete, metals versus tree bark and rocks. And so based on Kristen's work, we know these toe pads are bigger. And this has happened recently. This has happened within hundreds of years. And so we know they're bigger in urban populations. So what Bailey was working on was asking, how are the toe pads getting bigger? Is this just simply isometric change? Everything is just scaling up so that they have more area? Or are the toe pads getting wider more than they're getting longer? Or are the toe pads getting longer more than they're getting wider? Because this would have kind of kinematics repercussions for how they use their toe pads, how they run. How are they getting that more area? Where is it coming from? And so again, she used geometric morphometrics, outlining the shape of the toe, also outlining the lamellae, the scales on the bottom of their toe, because we also know these urban anoles also have more lamellae. They have more scales on the bottom of their feet, and their toes are bigger. And so is that, again, just an isometric issue that you have a bigger pad so you can fit more scales, you can fit more lamellae, or how are things changing? And so she did geometric morphometrics, and she also extracted just straight up linear measurements, toe length, toe pad width, toe length, toe pad length, and lamellae width, the width between each individual scale on the bottom of their toes. And so we did this for almost 250 species. We had a handful of urban populations and a handful of forest populations. And really the cool thing, we had a bunch of cool correlations, but the main takeaway is that they're primarily increasing in length. They're getting bigger in all their dimensions, but the majority of their change is coming from length. So the toe pad itself is getting a lot longer than it's getting wider, and the distance from the bottom of the pad to their palm is getting less. So the toe pad itself is taking up more of their toe. And so that's how they're getting bigger pads, which is really interesting because urban lizards also have to run a lot more because there's a lot less distance. There's a lot more distance between safe places to hide. And so a lot of times when we see toe pads in more terrestrial species, their toes are reduced. Their toe pads are reduced and kind of pushed towards the tip so that they can lift that part of their toe and still run. But here we're kind of seeing the opposite pattern. But urban anoles have to run a lot and have to cling to stuff that's hard to cling to. So we could imagine, like, it's really cool. And so now how do they run with these big giant toe pads is the next question, right? So there's lots more interesting things we can ask kind of going forward. Any questions so far?
Nope.
No? Cool. So we've talked a little bit about how things are shaped. We've talked a little bit about how things are shaped in relationship to the adaptation. So the last thing I want to talk about is cedal morphology or hair shape, and kind of go down another project that I'm working on down that road. And so we know the hairs are different across species. We know these hairs are different even within a single toe, but it's not super well studied. It's kind of studied at a real general level. But one of my pet hypotheses is that texture really has to be driving this. Texture with how the whole toe pad might be interacting with the surface, but also texture on how individual hairs might be interacting with the surface, texture on how the tips of individual hairs might be acting with the surface. You could imagine that texture at a nanoscale might affect how well each individual tip can attach. Texture at a micron level might affect how deep into the valleys the hairs can reach. Texture at a millimeter level might affect what percent of your toe can even contact the surface. So texture is something I'm really excited about, and I'm trying to figure out how to study it. And so part of the challenge is that even to measure hair shape just in general, these things are so small, we have to put them usually into a scanning electron microscope, into an SEM. That's the kind of the standard way to do it. And if you're not familiar with an SEM, you've got an electron source, shoots an electron down that you can focus with magnets. It hits the sample. The electrons hitting the sample bounce off or eject other electrons and then those electrons hit a detector, and you can draw a picture based on the stuff hitting the detector. That's actually really similar to how our eyes work, right? Our eyes, light hits stuff around us, bounces off, and then goes into our eye. This is different than a lot of your other light microscopes where the light goes through the sample and you can draw a picture based on the light that makes it through the sample. Or a transmission electron microscope works the same way. It shoots an electron beam through a sample and you can draw a picture on the other side. This depends on how things bounce off, the same way our eyes do. And the drawback of that is that your pictures, your images can get really distorted because of perspective. As things are farther away, they change size. And so unless you're looking at something perfectly perpendicular, if you're looking at something at an angle, you're starting to distort its size and its shape, which SEM suffers from. And so trying to draw morphological tape, trying to extract morphological measurements from a structure that you're never quite sure what angle you're looking at it at is problematic. And so our CETL measurements are we always have to take them with a grain of salt because of just the optical distortion that's built into SEM. And I didn't like that. That was bothering me. And so I started playing around. I started this in graduate school. I started playing around with other alternative ways to measure these things, to image these things. So I started playing around with confocal microscopy, which you can do 3D imaging. And I kind of got some images. I also played around with micro CT scanning, which I also got some images. But both of these, confocal microscopy and micro CT, are really limited to size. Confocal microscopy, it depends on what wavelength of light you're using, but you can't get much smaller than wavelengths of light. And as we already know, some of these structures on these hairs are smaller than that. Micro CT, again, you're limited by detectors and that kind of stuff. And so for both of these approaches, the best resolution, the best 3D resolution I was able to get was about half a micron, that's why you get kind of these globby 3D reconstructions, because we can't extract much more detail from that. And so then I started digging around for nano CT, which is a thing. You have to go to a particle accelerator for, though. And so I spent quite a while trying to get images out of the particle accelerator at Argonne National Labs outside of Chicago when I was in Michigan. And we got some images that sort of worked. But again, the challenge is now we're dealing with stuff that's so small. Is it vibrating or is it being deformed by this beam, by the electron beam, by the energy beam? I guess it's an X-ray beam. And so the reconstructions were really difficult because we had either movement and or distortion. And so because these scans took like 8 hours to do. And it was really tough getting into a Department of Energy research facility every time you want to like tweak your methods. So maybe it would work, but it was just really tedious to try to dial in. So what I've been working on now over the last couple months is using photogrammetry and using scanning electron microscope images. And so if you're not familiar with photogrammetry, what happens pretty much is if you take images of a thing from a whole bunch of different angles, you can reconstruct a 3D image of it. And so that can take into account optical distortion. And archaeologists and paleontologists use this a lot to build 3D reconstructions of their dig sites. Some folks out there have used SEM images for it. And so it's not completely crazy to use SEM images to do this photogrammetry 3D reconstructions. So we can put a chunk of these hairs into an SEM, take an image, rotate it, take an image, rotate it, take an image, rotate it, and we can build a 3D reconstruction from a whole bunch of these 2D SEMs. And so that's what I'm working on now. And I've got some preliminary images of 3D reconstructions based on these 2D SEMs. And so that's kind of what I'm working on right now. And I'm hoping to present that soon at SICB actually in January. And so we've got some preliminary images. Yeah, and so what's going to be cool about this with the 3D reconstructions using photogrammetry is that not only will we be able to start comparing species in a way that I think is a little more trustworthy, And so we're able to get around that distortion, that optical distortion from SEMs. We'll be able to start comparing different parts of the toe, but we'll also be able to start comparing textures. And so the last couple of field seasons I've done, I did field work in Australia a couple of years ago. I was in Hawaii a couple summers ago. I've been taking casts, molds of the different textures I'm seeing geckos on. And when I was in Michigan, I was able to use work with the engineering department and reconstruct two-dimensional surface profiles from these textures. So this is a 2D profile of like tree bark or a leaf. But with the photogrammetry, we'll be able to get 3D reconstructions of the surfaces they're using, which will be really cool. And so I'm really excited about that. And once we get either 2D or 3D reconstructions of surface texture, there's a ton of different definitions of texture we can start to extract from those and start to either correlate or find causal relationships, biomechanical relationships between hair length, hair density, hair stiffness, hair curvature, how sharp is your surface, how smooth is your surface, how spiky. There's a ton of different ways we can kind of break down texture and try to correlate it with hair shape or hair performance. So I'm really cool to kind of start getting into the nuts and bolts of the hair biomechanics in response to or in relationship with texture. And so I think that's kind of it. We kind of bounced around and covered a little bit of everything, how things are shaped and how that might be related to adaptation and some of the mechanics of how the hairs work. And so I already see one question up in chat, but if there's any other questions, I'm excited to take them. So the first question that I saw is, are there habitat materials that are especially amenable to van der Waals forces? Do anole toe pads also use van der Waals? So as far as we know, anoles probably use van der Waals. The tips of gecko hairs are a lot bigger. And so how van der Waals, nobody's dug into anole cetal biomechanics that much yet. We just assume they work like geckos. And as far as habitat materials so pretty much everything works pretty well with van der Waals. Everything is capable of producing van der Waals forces, except materials that hold on to their electrons really tightly. So like Teflon doesn't like to share its electrons. And so that's why things don't stick to Teflon. So geckos have a really tough time sticking to Teflon. They don't do really well with bare metal. But most natural surfaces like rock and tree bark and leaves and stuff like that, they stick to really well. I hope that answered your questions, Cliff.
So what's the extent of variation in adhesiveness among different species? Is it a really wide from some that are really not very sticky, quote unquote, to some that are just really strong in terms of being able to adhere to something?
Yeah, yes, there is. And so I want to say there was 3 or 4 different genera that toe angle test. So if you fall off at a really shallow angle, that might mean you're really good at generating friction, but as soon as you have to generate some adhesion, you probably fail, right? And so that angle that you're able to stick to kind of is a illustration of how much adhesion you can produce versus how much you're just good at producing friction. Some species can ride that glass up to 45 degrees, which means they're able to produce 50-50. Their maximum capability is 50-50 friction to adhesion, which is a lot. Whereas some species, as soon as that thing hits like 10 degrees, they're falling off, which means they're probably good at climbing vertical surfaces like anoles. So anoles you'll never see on fully inverted on a ceiling because they just can't produce that. They can't produce that much adhesion. They're really good at friction, but not so good at adhesion. And there are geckos out there that are really good at friction, but not adhesion too. And so if you've ever had like a pet crested gecko, they're really good at friction, but not so good at adhesion. And so there are geckos that overlap in performance with anoles, even though they're morphologically so different.
Hey, Travis, I have a question for you. That was a great talk.
Yeah.
It was really
Thanks.
cool. So is there any interaction between the toe musculature and how much adhesion and friction that the geckos can actually produce?
Probably, but nobody studied it. And so there's Tony Russell is an awesome guy. He just recently retired from Calgary. He's kind of the grandfather of old school anatomy of toe pads. And so geckos, that's one way we can tell who never had toe pads and who secondarily lost them, is so geckos can hyperextend their toes. It's one way they peel their toes off. And so they have muscles and ligaments and tendons and everything that let them peel their toes upwards. Some geckos don't have that. And so our conclusion is that those groups probably never had pads if they don't have the vestigial musculature to hyperextend their toes, versus there are some geckos that still have that even though they don't have pads. Anoles don't have that musculature, so they can't hyperextend their toes, but they have pads. So there's some relationship between that. Geckos also have blood sinuses under their toe pads. So there's something going on affecting maybe how soft the pad is kind of underneath. Geckos also have, so you know how you have tendons that go from muscle to bone, you have ligaments that go from bone to bone. Geckos have something like that goes from bone to skin. So their skin is directly attached into their bones. So they have a skin to bone tendon ligament thing.
Weird.
Super weird. Yeah. And so, but then on top of that, gecko toe pads still work even if the animal's dead. You can still plop a dead gecko and pull, and the hairs still operate. So it doesn't need muscles, but I'm sure that they have to be able to articulate. I'm sure they have a lot of fine control, but what that means, I don't know.
Huh, that's awesome. Okay.
Yeah. So I'm sure there's cool stuff going on, but we need, in essence, you need somebody to do some high-speed video of them running, which is hard to do, right? And then you can see them like hyperextending and attaching and stuff. Yeah.
Has Tanya Shea tried to do that with geckos, or is she only working on anoles? But that's who I can think of as like high-speed running with some stuff.
Yeah, that's totally up Tanya Shea's alley. That's up Tim Himes' alley.
Yeah.
That's up it's those guys that would totally do it.
Okay, awesome. Cool. Thanks.
Yeah, you're welcome. Tyler had a question. Do geckos press their bellies or tails down to help stick like some salamanders do? Also, I love the ICP reference. Yay! Not everybody gets the magnets joke, so that's awesome. So the cool thing, to even throw a bigger wrench into this, there are 3 or 4 genera of geckos that also have adhesive toe pads on the tip of their tail. So not only do geckos have adhesive toe pads, some have adhesive tail tips where they have hairs on the tip of their tails. So that's evolved multiple times in geckos too. So some have prehensile tails and they can generate presumably adhesion and friction with the tip of their tail. Yeah, it's super cool. And there's also, so folks have looked at, so geckos can detach their tails and regrow them. Anoles can too. And so how much force you start applying to your tail gets weird because there's fracture planes and all sorts of stuff. But some of the folks trying to develop gecko-like robots, so synthetic adhesives on robots that can climb, those engineers found out pretty quickly that geckos use their tail as not necessarily a counterbalance, but as they push their tails against a vertical surface to help keep the front of their body down so they don't kind of peel away from the surface. And so they had to start adding tails to the gecko robots to keep so that they didn't lever away from the surface. So I'm sure they use their tails for climbing and stuff. Yeah, but it's super cool.
Hey, Travis, I had a quick question for you, just kind of thinking of the phylogenetic context of understanding these traits. We often think about modeling, trait on a phylogeny and like fitting an Ornstein Uhlen back or, Brownian motion model. It sounds like with all this stuff that you're saying about, there being a skin to bone connection about, the spread of the digits, it sounds like modeling this in a single trait context phylogenetically might be misleading. And we might need to look at methods to model suites of traits. I was wondering what you thought about that.
Yes, 100%. Especially when you start to blend that with the ancestral state reconstructions, which if we just categorize things as present, toe pad presence versus toe pad absence, we get, because there are papers starting to come out looking at even species or species complexes within a genus where some populations / species don't have pads versus some kind of look like they do. And so even just trying to categorize presence versus absence of toe pads gets sketchy. And then if you would, depending what type of ancestral state reconstruction you do, if it's a threshold versus an MK, you get different results. And if you do an ancestral reconstruction with just geckos versus just lizards, you get different ancestral states. And so that's part of the challenge. Yeah. So I worked with Josef Weta, and we used an OU model. We used a Brownian motion, Brownian motion with a trend, and an OU model to model performance. And it looked like for anoles, they were a really tight OU on performance versus geckos were probably Brownian motion with a trend. But that may have been because we were assuming a single origin in geckos. So you got to get the ancestral states right before you can start fitting models, because then it pulls on your models, right? But
yeah, it sounds like there might be an interaction where, you know, you look at these other associated morphological characters, and if you fit a model to each of them, you might end up with that problem where at the ancestral states you have like an implausible set of characters at the node.
Yeah.
Tricky.
Yes, all of those. And I'm not, I can use those kind of comparative methods, but I'm not good enough to actually write them. And so, I think there's cool stuff we can do with more intelligent modeling, like threshold ancestral state reconstructions and kind of hidden state kind of stuff. But yeah, I'm super excited to try to figure out how to do those things. Any questions from undergrads? No?
You're shy. All right, guys. Well, let's thank our speakers. That was an excellent talk. Thanks so much, Trevor.